Let me just say thank you so much to ARM for inviting us here. This is our first year participating in this conference, and it really is just an honor to be among our colleagues and peers in the space who are doing such great things to drive medicine forward. It really is an explosion of technology, and we're glad to be a part of it and excited about where the future lies. I will refer you to our safe harbor statement. I will be making some forward-looking statements here today. So Amicus, as you may know, has a rich history in developing therapeutics for rare and orphan diseases. That's our mission. And our vision that we set forth last year is to treat 5,000 patients living with rare diseases with an Amicus medicine by the year 2023. And at the time, when we only had about 200 patients on any kind of Amicus medicine, that felt like an ambitious vision. This year, we have the goal of treating at least 1,000 patients with our approved drug Galafold for Fabry disease. And so we feel like we're well on our way, and I'll talk a little bit more about how we'll get there. But we do have a great platform that we've built. We have 600 employees around the world in multiple offices. We're present in 24 countries with Galafold. We have a phase 3 product for Pompe disease in development, which I'll talk more about. And last year, as a colleague we were speaking with the other day mentioned, we sort of jumped into the deep end by getting into gene therapy in a real way. We now have over 50 products between discovery and development in gene therapy. So let me start with Galafold. It really is the foundation of the company. It's what the company was started on. It's a chaperone, a pharmacological chaperone designed to bind to and stabilize misfolded enzyme in Fabry patients with specific genetic mutations. And I think that it's important for us to remember that many of the companies here started focused on regenerative medicine, but there's a whole host of potential innovations for patients living with rare diseases, and I think Galafold is a great example of that. We're hoping to do 160 to 180 million in sales this year. Today, the global market for treatments for Fabry disease is 1.4 billion. We are approved around the world, as I mentioned before, 24 countries with pricing and reimbursement. And what's unique about Galafold is that, again, it's indicated for patients with specific genetic mutations, and we've been able to work with regulators to, as we go and characterize new mutations, and there's new mutations being found all the time, rather than actually going back and studying those patients, we're able to just update our label electronically through the process in Europe and the United States, and we now have over 348 amenable mutations in the label. It was 260 when we first were approved in Europe in 2016. So a really exciting product, making, we think, a meaningful difference for patients around the world. And that really serves as the foundation of Amicus and who we are. The next program I'll talk a little bit about before I dive more into the gene therapy programs is our Pompe program. It is actually a combination of a enzyme replacement therapy, ATGAA, plus a chaperone. What we found was just as we can bind to and stabilize endogenous proteins, we can also bind to and stabilize exogenous proteins. And this is a really unique product in the sense that we knew, as many do now, that part of the key to success with protein therapies is they have to be taken up into the right tissues. So we worked carefully to select a naturally occurring cell line that produced that produced GAA with a high degree of carbohydrates, mannose-6-phosphate in this particular case, and then designed a manufacturing process around that cell line to ensure that we could preserve those key quality attributes. A number of the lessons that we've heard talk about over the course of the last couple of days in terms of the keys to manufacturing for gene and cell therapies apply here as well. And to give you a sense, we'll probably invest 100-fold more in the manufacturing for this program as we will for the next program I'll talk about for CLN6. So this is a heavy endeavor. That being said, the data we've seen we think is remarkable. We've shown data now in over 20 patients with Pompe disease, many of whom were switched from standard of care and came onto our treatment regimen. You probably are familiar that about 25 meters is a standard accepted clinically meaningful endpoint for or improvement for 6-minute walk test, which is the endpoint we're looking at here. And we've seen actually a 50-meter improvement out to now 24 months, both in naive patients, also in switch patients. And we think this will have the potential to be the standard of care for all patients living with Pompe disease. We're very excited about the program. We're in phase 3 development. We hope to finalize enrollment by the end of this year. So let me switch gears now and talk a little bit about gene therapy. And this really was a natural evolution for us. As I mentioned, the chaperone therapy is focused on stabilizing naturally produced enzymes. The protein replacement therapy, the enzyme replacement therapy, is focused on stabilizing and targeting externally produced enzymes. And with our gene therapies, we're looking at making sure we can stabilize and target internally produced enzymes. And so for us, this was really a natural evolution. I think there's another piece of it too, and I think Jeff Walsh and others have talked about this, which is this need for constant innovation. We have, as part of our belief statement, a duty to obsolete our own technologies. And so I think this evolution on this slide is a very real example of that. And I'll get to in a moment that we're already developing a Pompe gene therapy before we've gotten our Pompe enzyme replacement therapy submitted and hopefully approved. So I think that constant innovation is part of our duty at Amicus, and really a lot of what all of us are focused on in the room here. When we got into gene therapy, we did not have any technology we were focused on already, and I shared some of this in a panel earlier today. So we were free really to look out there and ask ourselves what we wanted to be a part of. So we really started with the diseases and the patients and the areas of highest unmet medical need. And there were two places within our kind of home of lysosomal storage disorders and rare diseases that made the most sense to us from an unmet need. The first was in CNS disorders, and the second was in neuromuscular disorders. And so we then narrowed that list down to about 100 diseases and then asked ourselves what were the best technologies available to be able to target those diseases. And we narrowed that down further and really brought together a portfolio based on two different partnerships. The first was an acquisition of a company called Selenex that brought our Batten franchise. Actually 10 CNS disorders. It came out of Nationwide Children's Hospital. And I'll show you actually some really exciting data there from our phase 2 study in CLN6 Batten disease. And the second partnership was with Jim Wilson, Dr. Jim Wilson at UPenn and his gene therapy program. And there we initially focused on Pompe disease and a handful of other indications, and then actually very quickly broadened that partnership once we saw the initial proof of concept data from our Pompe program, which I'll share a little bit here. And the real difference there is, Dr. Wilson continues to innovate around novel vectors, and that's such a critical part of how we can deliver these therapies effectively. But what we really bring to the table is a fundamental understanding of protein trafficking and the importance, again, of making sure that whatever it is you're delivering into the system, whether it's cross-correction or into the circulation or whether it's direct injection, that those products, those gene products, actually have the appropriate targeting mechanisms, and I'll show you some of that here in a minute. So many of you are familiar with these kinds of diseases, and again, part of the conversation over the last couple of days is just how much unmet medical need there still is out there. So Batten disease is a CNS disorder. It's caused by a mutation in the CLN6 gene, which leads to lysosomal dysfunction. There are over a dozen forms of Batten disease. But CLN6 in particular is a childhood form of the disease, so these children are typically born normal. Within a couple of years of life, they begin to develop gait abnormalities, speech abnormalities. By the time of their late childhood, they will have lost the ability to walk, to think, to speak, to see, and they typically pass away by the end of the first decade of life. So really, really devastating disease, no available treatment. And a father with two children with CLN6 Batten partnered with Dr. Kaspar at Nationwide Children's Hospital to develop a therapy, and that's really what formed Selenex, which was the company that we acquired. So really devastating disease, proven approach in other areas, in SMA, for example, with an intrathecally delivered AAV9 gene therapy. Same approach used here. And we announced this data just a few weeks ago, actually, the first data readout from the phase 1/2 study of CLN6 Batten, and let me share some of that with you here today. So if you're not familiar, what we're looking at here is the Hamburg Scale. So it's a 4-domain scale, but this is the language and motor functions. These were the endpoints that were used for the approval of Brineura, which was a protein replacement for CLN2 Batten disease. And when we first rolled out this data, I think many of the people, our stakeholders, didn't really appreciate how fundamental these data are. So what you're seeing here is organized from top left to bottom right by age of treatment. And so the youngest patient is on the top left, and the oldest patient when they started treatment is on the bottom right. And what you can see first off is we have stabilized this disease in these patients out to 2 years. And I think sometimes when we say stability, people think about, is that really that big of a deal versus gaining a milestone or being able to walk, which is, is obviously fundamental. But this really is profound. The natural history suggests that these patients would be losing 1 to 2 points on this Hamburg scale between these two domains per year. And so you can imagine after 2 years, these lines should be going, directly down. And I'll show you some natural history comparisons in a minute. But the other thing that jumps out at you here, and this is something we all know, the earlier you treat these patients, the better chance you have of having a significant impact on the disease. And even though we think, if you look at patient 1 and 4 who are on the bottom row there, we think we are having an impact and that's meaningful. Clearly, maintaining these patients on the top row there and the one on the bottom left, maintaining that at a full function, 6 when they start treatment, or 5 is critical. So finding these patients early, treating them early is critical. But let me show you what I think is a more compelling view of this, and this is what I mean by profound data. So the patients in red here are actually siblings from a natural history cohort that was being compiled leading into this study before the availability of the gene therapy. And unfortunately, these siblings here had already contracted the disease and developing symptoms before the availability of the gene therapy. And you can see they rapidly decline without treatment. And most of these patients are going to expire within 3-4 years of developing symptoms. What you can see in green are the treated siblings from this study. And clearly you see that the introduction of this gene therapy makes a meaningful difference for these patients and something that gives us great hope for the future. It's a phase 1/2 study, 12 patients fully enrolled, so we'll see the full data set here when the last patient gets to that 12-month endpoint. Our strategy is that we could use this combined with natural history to pursue registration. We still need to talk to regulators and confirm that strategy, but our hope is that we'll be able to pursue that. And I think folks heard Dr. Marks talk earlier about some of the ways that the FDA is partnering with industry to find faster paths to approval. We very much support that kind of strategy. In the last couple of minutes, I want to switch gears and talk a little bit about the Penn Collaboration. So again, started off with a handful of indications, then expanded rapidly. Includes primarily lysosomal storage disorders, but also includes some larger indications like Rett, Angelman, myotonic dystrophy. And again, the concept here is that we're marrying Dr. Wilson's technology around developing new vectors for better delivery, targeting, tropism, et cetera, and our technology around protein engineering. And I think even on the big screen, the font here is incredibly small, so you just have to take my word for it when I walk you through this. But effectively, what was an aha moment for us is that we realized that the proteins that were being expressed by these gene therapies were not optimally designed. And I'll show you some data in a minute that illustrates that perfectly. But if you think about the protein coming out of your manufacturing systems, if you think about classic enzyme replacement therapy, all of the work that goes into selecting the right cells that have the right structure, the right key quality attributes, effectively, if you're turning on an organ in the body to elute these genes into the system, you don't have that quality control mechanism. And so we want to make sure we can do everything we can to preserve the quality of that protein so that it can then get up into the tissues where it's meant to be used and catabolize substrate. So these were data that we showed actually at ASGCT earlier this year. And it's a little hard to see with the colors, and I apologize for that. But effectively what we were looking at is knockout mice. On the left-hand side, you can see a vehicle, so no treatment. And that dark purple staining is substrate glycogen in the CNS. So this is in the spinal cord. The next slide there is the naturally produced GAA gene therapy. So this is an unmodified protein, and you can see very little impact on the glycogen in the spinal cord. The next panel, that lighter purple, you can clearly see a reduction in that glycogen, a significant reduction in that glycogen. That is our engineered protein delivered via gene therapy, and it's very similar to wild type. So we think that marrying of those two technologies is going to be really exciting and will differentiate us from many of the approaches that are out there and hopefully have a better impact for patients. We hope to declare a clinical candidate by the end of this year. And then we have great partnerships with CMOs. We've talked a lot about that this year in the conference. The last thing I'll leave you with is, we firmly believe at Amicus that our goal is to treat as many patients as possible. We think that the disease is the competition. We think, of course, we're biased. We think our approach is the better approach, but it honestly is inspiring to have so many people here in this audience working to affect the lives of these patients and the patients that we're all focused on. So we applaud your efforts. We're looking forward to continuing on the journey, and I thank you for your time.